when should we expect a clinical update from the pivotal sickle cell and thal programs later this year?
Um, at a 20,000-foot level, Tyler, I'll just start off by saying that we're very pleased with how things are shaping up for 2022 for the company. We have multiple of our franchises moving forward in the clinic. This is the hemoglobinopathies franchise for sickle cell and thalassemia, our immuno-oncology franchise, and now our diabetes franchise is also in the clinic. And we hope to talk a lot more about our in vivo programs later this year as we attempt to bring more than a few of them to the clinic in the next 18 months. But overall, I think we're very pleased to be in a position where we could be one of the first companies to file a BLA for a CRISPR-based therapy in the world. And this is with our CTX001 program, where we've seen remarkable data for this drug in both thalassemia and sickle cell patients. We'll certainly provide a data update this year. I think our last data update was for 22 patients with quite a bit of follow-up, but we'll have a greater number of patients and a longer follow-up as we get closer to hopefully our filing timeline at the end of this year. And I think the program continues to move along really nicely. The trials are fully enrolled. We've made a lot of progress in our ability to dose at scale throughout the last couple of years, together with our partner Vertex. And at this point, Vertex is leading the efforts on getting ready for a commercial launch and all the prep work that goes with enabling commercialization of a drug. So all in all, I think we feel well positioned and also believe that our competitive position continues to strengthen as we go along.
A very helpful overview. Can you just remind us what's the latest, if there's an update on enrollment for the program, or if it remains the same per your last update?
Yeah, as we disclosed in one of our updates earlier that we fully enrolled the trials. Just to remind everyone, both CLIMB 111 and CLIMB 121 trials for thalassemia and sickle cell respectively were designed to enroll 45 patients each, and we've completed enrollment in both trials. We also commented that we had dosed over 70 patients. Now, if you look at past precedent and other rare diseases where they've reached BLA filing or approval, given the type of data we're seeing, we don't even know if you need 45 patients worth of efficacy data in a filing package. But I think the trials were designed to 45 patients. I think we'll have a safety package that spans both thalassemia and sickle cell because essentially the same product. And I think that should get us to a robust enough data set that hopefully enables approval.
Got it. And I guess related to the efficacy data required for filing, what duration of follow-up do you ultimately think is needed for sickle cell and thal BLA filings? And, I guess you're planning to file both of them hopefully by year end, definitely thal, but what is the potential risk of having to separate the two?
One of the advantages of having RMAT designation, and these are companies that have RMAT or breakthrough designation, is it changes the way you work with regulators around as we get to a filing package. To investors and to Wall Street, it may not seem like much, and which is why I think the RMAT designation carries a lot of weight operationally. I think for our CTX110 program, when we received the RMAT designation, I don't think it probably escaped the attention of many. But I think for us internally, it was an incredible deal to be able to avail of that pathway and the support of regulators. We found regulators to be very constructive both in the US and in Europe. And I think as we discuss both the clinical and non-clinical aspects, we're converging on all the requirements, whether it's the exact number of patients in the package, the exact file, the duration of follow-up on the patients. And we believe that we've dosed enough patients to meet that bar. And then on the non-clinical aspects, I think, while we designed the entire process to be commercial ready from day one, which is one of the advantages we have of designing it de novo versus inheriting it from an academic clinical trial, I think there's a lot of little details that need to be ticked and tied. And I think we're getting to that, and we'll be converging on all those aspects throughout the year as we get to our goal of filing by the end of the year.
Okay. And you mentioned your partner Vertex, clearly a heavyweight in the biotech space, so an incredible amount of resources that you'll have available through them. What learnings have they potentially taken away from Bluebird's early experience with beta thal in Europe? Is there, and as you think about launching in the US market, so, anything you guys or Vertex plan to do differently, or what's the focus?
Yeah, I think we have full confidence in Vertex, who have tremendous capabilities when it comes to commercial preparation, but also experience in the rare disease space working with the entire ecosystem, not just physicians, but the centers, the treating centers, the payers, um, the uh, the regulatory agencies globally, and I think that's all going to carry a lot of weight. I think the strength of the package is undeniable, and if you look at the relative benefit that these patients are getting, from a CMS publication not too long ago, there was data that showed that for sickle patients, um, they had the average number of VOCs in their Medicaid population was so high, and the patients ended up in the hospital so many times that the cost of care for even one of these patients is well north of 250K, closer to $300,000. And I think if you're taking away all those costs from a pharmacoeconomic standpoint, delivering tremendous value to these patients in making them normal and productive citizens of society, I think that's an undeniable argument to enable for the system to enable access for all these patients suffering from these diseases. By the way, which haven't had any sort of meaningful intervention for decades. One of the longest known molecular diseases out there. And I think there's obviously a lot to learn from not just Bluebird's experience, but from other launches for gene therapies or cell therapies, not just in the U.S. and Europe, but other parts as well. And we'll be applying all those learnings. But in many ways, I think the strength of the program, or the strength of the data from CTX001 will speak for itself.
Okay. If you think about the launch of OxBright Up, some might argue that it underperformed expectations, and that there's some patients that expressed a lack of desire for treatment. Do you feel that there's read through from that to the CTX001 launch, or ultimately are you going after a very different, more severe patient population based upon that 250K number that you cited?
Yeah, I think it's the patient population is very different. I think you're talking about serious sickle cell patients. I think if it's this is a patient population that's unique. But at the same time, I'll tell you, we've had direct interaction with some of these patients. In fact, one of them was an employee at CRISPR who, if you have 4 or 5 different VOCs per year where you end up in the hospital and any one of these episodes you could die, that's a scary life to live. And in fact, some of these patients, when they show up in the hospital with debilitating pain, they're often confused for opioid addicts, and they don't get painkillers. I think for these types of patients who are living with pain day in, day out, I think you're not talking about the same paradigm of, are they going to be compliant, or are they wanting these therapies? They are seeking something to get them out of this life that they live with a lot of pain. In fact, there's a recent article in BBC about one of the patients that were enrolled in our trial and how life-changing and how dramatic the intervention was. And I think for these patients, it's a very different dynamic than the dynamic that GBT and OxBright may be facing, which is the less severe end. And also you have to put it all in context of what change is the patient expecting? How dramatic an impact is the patient expecting? So I think very different dynamics. And I'm confident, in fact, that just judging from the interest in our clinical trials and the number of patients that want to be part of clinical trials, that there's going to be a lot of demand here. And that's changed, by the way. When we started the trials, it wasn't, people were still trying to understand what gene therapies were, what it meant to have your hematopoietic stem cells, replaced or corrected. And I think all that's changing over time. But right now, I would say that there's tremendous interest in our therapy, both from a clinical trial perspective, but just generally.
Yeah, and I was previewing the results for the non-malignant hematology panel, and the awareness among physicians for CTX001 is definitely high. I was impressed. Okay, so maybe a last question with 001. Your efforts to pursue a milder conditioning regimen, what's the approach, and ultimately how close is this to becoming a reality, since people have been working on this for quite some time?
Yeah, we're pursuing, this is one of the bigger priorities for us, or top 5 priorities for us as a company, is to come up with a gentler conditioning agent that not only expands the addressable patient population, but also changes the trajectory of penetration of the market with CTX001. And we're pursuing both antibody as well as ADC approaches. You can ask the question, why not just access something that's out there in the market? And there's all these companies that are developing these conditioning agents. The difference is these companies are developing agents typically for cancer settings, particularly AML or GVHD settings. And the PK/PD profile that we're looking for is very different. You want these agents to go in and kill the existing hematopoietic stem cells, particularly long-term hematopoietic stem cells, but then not stick around too long to damage the cells that we put in as the drug product. And that type of PK/PD profile is hard to hit, but has to be optimized for this specific application. So we're undertaking internal programs around both ADC and antibody approaches. And I think Vertex is doing the same. So we're confident that in the next 2 or 3 years, we'll have something that could be meaningful as a gentle conditioning agent.
That's great. Before we move to CAR T, I have a couple questions from the audience just on the recent IP ruling in favor of the broad, and just what's the specifically one person asked, what's the plan to pivot if the licensing agreement, if a licensing agreement of some sort needs to be completed? How expensive might this be, or what might a licensing agreement look like, and just in general, how relevant is it for your existing programs and business?
Yeah, I think the recent interference ruling by the PTAB, I think, sort of maintains status quo. I think, if I contextualize what was happening, it was an interference where CVC, California, University of California, and the Charpentier Group were challenging the validity of Broad's patents. This was nothing to do with anyone else challenging our patents. We already have 45 issued patents in the US, which range in applicability of use of CRISPR-Cas9 across all cell types in different environments. And it's a pretty broad set of IP that we have as a CVC group. What this ruling allows is that the Broad and its licensees retain some of the IP that we had challenged. Now, we do believe the PTAB got this decision wrong, and we have the ability to appeal. But that aside, I think, if you take the global equation, in Europe, we have a dominant position with our IP. And I'm alluding to the CVC group. A number of patents that the Broad had been issued have been overturned. Our patents continue to be upheld. So is the case with several of the other countries outside of the US. And the US, we have this situation where, CVC have some of the IP issued and the Broad has some IP issued. All said and done, I think what this may mean is there needs to be some potential cross-licensing opportunity. And it may end up being economics free or something on a relatively low burden. So from a business standpoint, I don't think it's something that we're that changes the direction or trajectory or anything else at this point. For us, it's business as usual. And we'll continue to see how the IP events may be shaping up. There's other interferences ongoing. There's other parties in the mix. It's very complex. And I think the media generally take a very simplistic sort of headline and put it out there, which generally I think doesn't capture the full picture as I've framed it.
Okay, that's helpful. Thank you. Maybe we'll move to CAR T. So regarding the 110 pivotal trial design, I believe you recently stated it was finalized. So can you describe it for us to the extent that you're willing to? And I think you said it could take about a year to enroll 75 to 120 patients. Is that still your latest thinking?
Yeah, so I think just to frame the picture, I think CTX 110 is our CD19 CAR T, where we showed some very interesting data in our initial data disclosure. I think 4 years ago, I remember this is actually around the time of your conference, Tyler, where we were meeting with some big pharma companies and they wanted to see our initial concept design for CTX 110. And I remember one of the more senior R&D leaders in big pharma saying, it's going to take a Nobel Prize for a patient to have a durable, complete response from an allogeneic CAR T. And here we are, where we have, a handful of patients who are cancer-free with a, single dose of CTX 110. And what we've also learned through the efforts, not just us, but some of our peer companies, is that doing a consolidation dose where you completely eliminate the cancer after the second dose will lead to a greater proportion of patients achieving CR and a greater proportion of the patients that achieve CR to have a durable CR. And with that notion, especially armed with the PK/PD data that we have around the effector to target ratio that we disclosed, we have great confidence in our design where we do 2 rounds of CTX 110 dose with Flu/Cy at dose level 4, which is 600 million cells. And we've begun enrolling in this pivotal arm at the beginning of this year. And we've enrolled a few patients already. And we believe this trial, as a single arm trial, could be registrational, but that's something we have to fully align with the regulators on. I think, again, having the RMAT designation helps quite a bit in getting those discussions going with the regulators in the US. And similarly, we find the European regulators quite supportive on an allogeneic CAR T. I think there is a major need for an allogeneic CAR T in Europe because the autologous CAR Ts have been hard to access and the price point is prohibitive for many of the countries in Europe. And I think we're going to get broad system support for CTX110. I think our competitive position, continues to strengthen as we enroll patients. And hard to say how long it exactly takes to enroll a patient. I think that, be the first to acknowledge that there's, way too many CD19 programs out there, and I'm sure investors are confused with all the CD19 CAR T programs or allogeneic programs, including NK cells and bispecifics and everything else out there. But I think we have this clear window to get to market first with an allogeneic therapy, which offers all the convenience that autologous therapies don't, and with a response rate and durability that's meaningful enough where this could be a $1 billion drug, and again at relatively attractive margins. So that's our strategy, and we continue to act with a sense of great urgency around the program given the competitive dynamic.
Very helpful. So a couple follow-ups on the redosing that you mentioned. I imagine that's going to happen around day 28 or a month later, and will that require another cycle of Flu/Cy prior to that second dose?
Correct. It's around a month later, and we've taken a conservative route to say that we will apply Flu/Cy conditioning again a second time. Over time, we may interrogate approaches where we don't have to do Flu/Cy conditioning the second time around, or toy with the smaller dose for the second infusion of CTX110. But at this point, we're going with the standard Flu/Cy conditioning once initially and once after 1 month. And all patients, whether they get CRs or PRs or stable diseases, will get the second dose.
Got it. Okay. And what about progressors? Will you retreat those, or how will you determine whether they're retreated or not?
I think there will be on progressors. There, I think it depends on the patients. I think there's a choice to redose a progressor, but in some cases the patients may not be able to tolerate a second Flu/Cy conditioning dose, or you may not see the benefit that you desire, and in that case it may not be beneficial to redose the progressor. But generally, I think this is all aimed at, what we're seeing is, if you can convert stable disease or PR to a CR, that increases that fraction of CRs that we have, and then if you can make the CRs more durable, I think that'll be a remarkable sort of outcome from this trial.
So we'll have to see how much redosing adds to efficacy based upon the prior update. But is the goal really to get to market as quickly as possible and then iterate the product with kind of next generation candidates moving forward to further improve efficacy?
Yeah, we have a 2-point strategy. I think we want to get to market as quickly as we can with CTX110 because we believe that not only will it have a strong place in the options that physicians have in relapse refractory or third line settings, but we can also move it into earlier lines of therapy. I think there's great potential for an allogeneic CAR T to leapfrog autologous CAR Ts. There's high-risk patients in frontline settings, for instance, where physicians are quite interested in applying an allogeneic CAR T. You could use allogeneic CAR T as a major debulking agent and then do maintenance with R-CHOP potentially. So there's all those options, given that we don't have the supply challenges and ability to do these trials quickly that we could interrogate in a capital efficient manner. And then we continue to iterate. I think this is going to be a space where we're already working on a Gen 3 design, right? We already have a Gen 2 program that we've locked in terms of design and edits. We're now designing our Gen 3 program, and having our own facility in Framingham allows us to iterate very quickly on the next gen versions. And by the way, the next gen programs are, again, there's a great capital efficiency here. If our first program costs us $100 million to get to an IND, the next gen program, I think, is less than a third of that cost because we have all the components that we desire already in terms of the editing and the setup that have a threshold set of costs. So I think what you're going to see is every 2 years a new version of the CAR Ts from our end that in most models at least are definitely a lot stronger, more cytotoxic and exhaustion proof. But we also don't want to confound and confuse the marketplace. I think so we'll have a cadence to how we bring the next gens on board.
Okay, so you'll have 120 and 130 phase 1 data by the end of the first half, 120 being the BCMA candidate and 130 being CD70. I have a client question that asks, for 120, what would you need to show to continue the program in light of the legend J&J or Janssen data or CARVYKTI?
Yeah, no, I think that's, the BCMA, each of these indications have a different competitive dynamic, right? I think BCMA is an indication where there's, even with the Janssen legend data and the availability of the product, right now, if you look at the Abecma, I think there's supply is a constraint. It's not demand. There are a lot of patients who want to get on CAR Ts or suffering from multiple myeloma that don't have access to it. That said, I think there's a tolerance around efficacy relative to autologous, given the convenience of allogeneic, but there's a limit to that tolerance. And the question is, if we fall below that tolerance limit, it becomes a strategic question for us of whether we try to leapfrog the autologous therapies with our Gen 2 program, or do we continue the fast follower strategy or first to market strategy with an allogeneic program. We do think that the edits that were made with our Gen 2 programs in mouse models are an order of magnitude better in relevant multiple myeloma mouse models. So those things have to all be put in context as we determine our strategy once we look at the data and think through that in the middle of the year. With CTX 130, I think we continue to be very intrigued by this target. It's a very interesting target, not just from an expression standpoint, but from a biology standpoint, in terms of the tumor expansion, the tumor profile that we see not just in renal cell carcinoma, but across a number of different tumor types. And we'll continue to evaluate the data. I think we've now established a redosing protocol in CTX 130 as well. And by the way, patients suffering from these tumors or these cancers literally have no other options. I think so. We're first in class with CD70, and it's a landscape where there's really no competition.
Should we, with CD70 and renal cell, obviously you're treating, I believe, both lymphoma and renal cell patients, but should we be expecting responses in renal cell patients and eventually some sort of response rate, or is it just too early to say that?
Well, I think that's the goal. I think in renal cell carcinoma, it's a very bleak disease if you have it, metastatic renal cell carcinoma. I think even the frontline approval of PD-1s, the CR rate was 10% or less. This is in combination. And so, the expectation around CR rates are low, given how difficult the disease is from a treatment standpoint, given what we've seen with other agents. But certainly the hope is you see responses and you see the potential of allogeneic CAR Ts in solid tumors, which I think many have questioned, but I think we do think that allogeneic CAR Ts are going to have a strong role to play in solid tumors.
Okay. Man, running out of time here, but I guess maybe we'll wrap up with one last question. What aspect of the CRISPR story do you believe is most underappreciated by investors, Sam?
Well, I think, just judging from this fireside chat and most of the discussions we have with investors, I think we have 4 franchises. If I think about my time spent at the company, I spend equal time on the 4 franchises: hemoglobinopathies, immuno-oncology, diabetes, and in vivo. We'll be talking a lot more about in vivo. We'll be talking a lot more about diabetes, but right now, a lot of the focus is on hemoglobinopathies and immuno-oncology, rightly so, because it's most advanced in the clinic. And if there's something to handicap because there's data we've already put out there. But I think, what we want to do is have each of these franchises get to a point where they become sort of self-sufficient. The hemoglobinopathies can be a self-sufficient in the black franchise for us relatively soon. And I think hopefully similarly with immuno-oncology, given CTX 110 has a path to becoming $1 billion drug at very high margins that can fund everything we do in immuno-oncology. And there's a lot we need to do. I think there is a bit of catch up we need to do on in vivo. But again, the data are impressive that competitors have put out there, which effectively tells you that the whole sector could be very meaningful in vivo. And diabetes is something that could get us to a $100 billion company if things work out. So we feel very good about where things are. I think this cool down in the market is actually the best thing that could happen to CRISPR, given our balance sheet. It helps us go on the offensive when others are being defensive, and help us consolidate capabilities and talent. And I think when we come out of it, it'll just allow us to accelerate a lot faster than others, just like we see in recessions in the past.
Okay, wonderful. Thanks very much for your time. I enjoyed our discussion, Sam.
Thank you very much.